Welcome to C&EN Uncovered. I'm Craig Bettenhausen. C&EN Uncovered is a podcast series from Stereochemistry. In each episode, we'll take another look at a recent cover story in chemical and engineering news and hear from C&EN reporters about striking moments from their reporting, their biggest takeaways, and what got left on the cutting room floor. Antibiotics are critical medicines and arguably the most life-saving class of treatments modern medicine has provided. But it's an arms race, with microbes constantly evolving to resist antibiotics and chemists looking for new ways to kill bacteria, yeast, and other pathogens. In this episode, we'll look at how genome sequencing, artificial intelligence, and synthetic biology are coming together to break log jams in antimicrobial discovery. We're here with C&EN Assistant Editor Max Barnhart, who recently wrote a cover story on the topic. There's a link in the show notes, or you can find it in the June 23rd print edition. Hi, Max.
Hey, Craig. Thanks for the introduction. That was great. In this conversation, I'm going to use antimicrobials and antibiotics often interchangeably. Antimicrobials broadly means stuff that could be effective against bacteria or fungi, antibiotics specific to bacteria, but you'll hear me use both terms throughout the conversation.
That is useful. I didn't realize that distinction. That's interesting. So for anyone that hasn't had a chance to read the story, can you give a brief recap of what's in there?
Yeah, totally. The story, it asks the question, are we in a new golden age of antibiotic discovery? There was an initial golden age of antibiotic discovery way back sort of between the 40s and the 60s, where scientists looking in microbes found different classes of antibiotics that we still use and appreciate today, like tetracyclines, cephalosporins, macrolides, aminoglycosides. These were all discovered between 60 to 80 years ago. But in the late 60s, the rate of antibiotic discovery dropped off considerably. And you could call it a dark ages almost, where we weren't discovering much of anything new. But over that period of time, pathogens started to become resistant to the antimicrobials that we were using. And now today, or rather just a few months ago, I reported on two different stories before writing this cover story. I reported on the discovery of a Pauline macrolide antifungal called Mandomycin, and then the very next week, lasopeptide antibiotic called Lariocidin. Both of those discoveries were published in Nature. Both of those compounds demonstrated some new and unique antimicrobial activity that made them effective against antimicrobial resistant pathogens. And so, from there, I had to ask myself, is this a coincidence or is there something more? And to quote my favorite character from Star Trek Deep Space Nine, Garrick, he says, "I believe in coincidences. Coincidences happen every day, but I don't trust coincidences." So I asked around, and yeah, it turned out that there was a trend here.
When one's discovering new antibiotics, how different does it have to be? I know that in narcotics, you add a methyl group here and there, and you've got, from the DEA's standpoint anyway, a totally different molecule. But is it iterations on the same couple things, or are we talking completely different modes of action?
Yeah, I mean, that's a really great question. And, I think it's really important to mention that when I talk about a golden age of antibiotic discovery and a dark age of antibiotic discovery, we're talking about the discovery of new classes of antibiotics, right? And exactly what defines a class of antibiotics is not super well organized. Like, there are different ways you can think about classes of antibiotics, but the way that I'm going to use it for the purposes of my story and for this conversation is classes of antibiotics are based on their structure and mechanism of action. So a lot of antibiotics were discovered over those dark ages that fell into pre-existing classes of antibiotics, right? A bunch of tetracyclines were discovered. That's not just one single kind of antibiotic. And those have a variety of effectivenesses, a variety of toxicity to humans. Certainly, among the classes of antibiotics that were discovered early on, we were able to iterate and get better and better, less toxic, more effective. But when it comes to these pathogens that can evolve resistance, it's easier for them to evolve resistance to an antibiotic that has a mechanism of action that they had already been exposed to in the past. So a riff on a tetracycline might be effective for a little bit, but it's maybe not going to have long-lasting ability to overcome that antibiotic resistance. The discovery of new classes of antibiotics and antimicrobials, like Mandomycin, like Lariocidin, these discoveries that were published in Nature earlier this year, because they work in ways that are totally different from what we have been using in the clinic, all of these pathogens that we want to use these new tools against haven't been exposed to anything with that mechanism of action. So they're much less likely to evolve antimicrobial resistance against those drugs. Hopefully, if these compounds do eventually make their way into the clinic, we can prescribe them and use them in really sustainable ways so that these pathogens don't develop antimicrobial resistance over time to these specific drugs.
Does antimicrobial resistance fade evolutionarily? Like if we stopped using something for a generation, in 20 years, would we be able to use it again? Or are we just creating permanently invulnerable superbugs with our antibiotic use?
Yeah, that's hard to say, right? I'm an evolutionary biologist by training, and we can look at trends, but it's really hard to predict the future. Certainly, if we think about not a bacterial infection, but if we think about, the COVID-19 pandemic, something that we're all familiar with, these different strains kind of arose, right? Each year there would be another few strains, and vaccines were maybe not as effective against the new strains that would pop up. And then those circulate amongst the population, get really high, and then maybe die down and another strain takes its place. That happens with bacteria and fungal infections too. And, certainly there have been outbreaks of things that we would consider to be like extensively drug-resistant bacteria. The real nasties. Those are really difficult outbreaks to deal with. But over time, they can wane, They're maybe not representative of the majority of these bacteria that are spreading amongst the population. Drug resistance takes energy, so when not exposed to these different antimicrobial compounds, maybe then it becomes more evolutionarily efficient for those bacteria to evolve less drug resistance so that they can grow faster or something along the like. But this is all theoretical, and not predictive. I would say clinicians are trying to use antibiotics more effectively these days to prevent more and more antimicrobially resistant pathogens from evolving. But it's a race, In evolutionary biology, we call it the Red Queen hypothesis, based on Alice in Wonderland, right? You have to run as fast as you can just to stay in the same place.
So a lot of the story is about these drug discovery methods, these antimicrobial discovery methods. Can you talk a little bit about the different methods that are in play now versus, the days of yore?
Yeah. So I think the big developments, right, in antimicrobial discovery have been genomics, right? So the ability to sequence DNA of pathogens and just of, the tree of life in general. We also have AI, which is emerging as a tool that can be used to explore the chemical space virtually. A chemical space which, there are more potential compounds to be produced than there are stars in the Milky Way, right? And AI can be used to help determine which of those potential compounds might have antimicrobial activity. We also have synthetic biology, which allows us to take the biosynthetic pathways for compounds that are produced in different microorganisms and engineer them into yeast or some other microbe in such a way that we can produce them at the really high concentrations necessary to be useful for manufacturing a clinically relevant drug. Broadly, those are the different things that are going on, but really the thing that makes all of this work is the ability to sequence DNA and sequence DNA of just the random microbes that you find in soil that often are producing a lot of these really highly effective clinically relevant compounds.
And then so part of what's really been the step change is the dramatic drop in the cost of doing that kind of sequencing. What kind of effect is that having? Because where was it and where is it now as far as if I have a bacteria and I want its genome sequenced?
If we think about, right, the Human Genome Project, which is around 25 years old now, we're talking millions of dollars, right, just to sequence the DNA of one human. Human genome is larger than that of a bacteria, but still, an enormous amount of money to do DNA sequencing. And over time, that has dropped down exponentially to the point where certainly around the time that I started graduate school back in 2016, you were talking about, maybe it's $1,000 to sequence the genome of a bacteria, right? Which is not that much money comparatively to that of the Human Genome Project. But then 10 years later, we're at the point where you can sequence the DNA of a bacteria for like $100, right? And it ends up being really high quality. And the amount of effort to do so is almost trivial. It takes half a day of work in a lab to extract the DNA and prepare the sequencing libraries, and then you send it off in the mail to a facility, and a few weeks later you get back the genome. And then you can do all of this cool analysis on it. Human genomics is a whole nother beast. And now we're getting into like personalized medicine and stuff, but they weren't doing whole genome sequencing like these researchers are doing with bacteria. Again, human genomes are larger than bacterial genomes, and the analysis and technology isn't there from like a commercial home use perspective to go and get your entire genome sequenced. That's just not efficient. It's technically possible and still relatively cheap. But when it comes to bacteria, which have these tiny, tiny genomes, it's just easy to bang out hundreds of them, right, for the purposes of doing one experiment to find one compound.
So back in the antibiotics, once they've found a compound of interest, looking at these genomes, looking at some of these mechanisms, that's great. But manufacturing at scale is a whole separate problem. How are they doing that?
Yeah, a lot of the folks that I talked to, the actual bioprospectors who are going out looking for these new compounds and trying to find something useful, their role kind of tapers off once you get to manufacturing and like actually developing these compounds for clinical use. I spoke with academics, right? And the pipeline of discovery that they described to me, is they'll spend years, 5-7 years trying to find a new compound. Halfway through that, they'll actually discover something, and then they have to test, against pathogens, against, a whole bunch of different cell types and disease types to see if it is effective, to see if it's toxic, right? Maybe they'll engineer the compound a little bit more to increase the effectiveness and make it better or less toxic. Then, maybe they try to discover the biosynthetic pathway in the bacteria that's actually producing this molecule, engineer that into something else, produce that at high concentrations. All of that goes into, publishing the Nature paper. Mm-hmm. After that, usually to get it into the clinic, it requires collaboration with folks that are more set up to do clinical trials. We're talking, big pharma often. Yeah. Or, they'll turn some of this into a startup. But when it comes to antibiotics specifically, these are not high-profit drugs for pharmaceutical manufacturers to produce.
I was surprised to read that in your story.
Yeah.
I think of these as being so effective, as you saw in my intro.
Antibiotics,
they're so powerful.
Yeah,
but
they are, right? But they need to be cheap because, unfortunately, where the majority of these antimicrobial-resistant infections are happening are in countries that don't have the established medical system that, we do in the United States, where people don't have the income levels that we do here in the United States or in other parts of the world. And, you can't sell an antibiotic for big money to someone who doesn't have the funds to actually purchase it. So there have been here in the United States some policies that have been talked about, like the Pasteur Act, which sets aside funds specifically for developing and manufacturing these critical need antimicrobials. But they haven't gotten much traction in Congress. It's pretty unlikely that that'll happen anytime soon. Organizations like the Gates Foundation have, funded development for a lot of these things, as sort of a charity almost. And that's probably the economic model that is going to need to be used moving forward to get these compounds ready for clinical use and then manufactured and distributed.
Interesting. So when we were pulling effectively molecules out of nature, these molecules do a lot of different things. Can some of these techniques, also be used to find solutions in other fields of chemistry? And are they watching these screens for, it doesn't kill microbes very well, but it's a really good, surfactant or...
Yeah, absolutely. I'm actually working on a story right now that will be published by the time this podcast episode goes out, I'm sure, about a compound discovered in fungi that it shows effectiveness at killing leukemia cancer cells, right, with low toxicity to other human cells, which is fantastic, right? We need more anti-cancer drugs. Cancer is a big problem. And when researchers discover a new compound that looks interesting in bacteria or fungi, they don't necessarily know exactly what it's going to do at that point. So they screen these compounds against bacteria, against fungi, against cancer cells, against other kinds of pathogens to see if they do anything at all. And maybe in some cases they show antibiotic activity. Maybe in some cases they show anti-cancer activity. Maybe in some cases they show some kind of activity that no one predicts but is useful for some really niche purpose. And but you have to test in order to figure it out.
One of those classic paradigms in this kind of research, in a lot of chemistry, is the structure-function relationship, which doing it analog, doing it manually, trying to do structure-function relationships is a long and painful process, and you can spend a whole career just figuring out how an electron transfers within a very specific molecule. Is AI changing that because it can eat reams of data and spit out, or are we not quite there yet with what AI is doing to this field?
AI is great. AI is super helpful for discovery of new potential drug compounds. There are still limitations, right? When AI needs to be trained on a pre-existing data set, so when it comes to chemistry, these AI models can be trained on compounds that we know already have antibiotic activity, and then be unleashed, to look into the chemical space and see if there's anything else out there that might have that antibiotic activity. But it can only recognize stuff that we kind of already know might work. And there's kind of a requirement to find and develop, without AI, new interesting molecules so that maybe those can be fed back into the training data and we can accelerate the rate of discovery from there. And, unfortunately, when these molecules are discovered, researchers can do some amount of figuring out exactly how the molecule might function. But there are also times where, something works and people don't know why, but even then, it's still worth exploring.
So as we discover these new avenues for drug discovery, is it easing the fear of antibiotic resistance?
People are optimistic that these potential drugs can work, right? It's going to take 10 years to get them into the clinic, or something around that time, because they have to go through phase 1, 2, and 3 clinical trials. There's a really high bar of effectiveness and safety that needs to be cleared before these things get used. But nobody's saying that we're winning the fight. It is going to have to be a constant. We'll discover something, we'll get it into the clinic in 10 years, and then hopefully there's a dozen other new drugs that are in the pipeline to get approved along the way, because we are going to need all of them. And I don't think we're ever going to get to a point where antimicrobial resistance is no longer a problem. We may get to a point where we have the tools to deal with it and we have the strategies to slow the rate at which antimicrobial resistance is rising, but it's going to be something that researchers and clinicians are going to be thinking about for the foreseeable future.
One of the things that caught my eye in your story was you talked about understanding the resistance mechanisms as one of the fronts of this. But at the same time, it seems like a lot of what the bioprospectors are doing is looking for these antibiotic mechanisms, these things that kill microbes. So how does understanding the resistance mechanisms lead to new antibiotics?
Yeah.
It's not quite the same thing, it seems like.
Yeah, understanding where resistance comes from kind of allows researchers to maybe hone in on molecules that they think are doing something else. Finding these things is actually really difficult because, as it turns out, bacteria and fungi, they make a lot of compounds that are effective at killing other bacteria and fungi. But a major problem is folks have already discovered those compounds.
Yeah.
And so using some of these advanced chemical and computational tools, people try to filter out that initial level of antibiotic activity that can be seen when you just break open a microbe and let its juices out, and see if it kills anything. So when you start to remove the antibiotic compounds that you already know are in there, that you already know have resistance in a population of bacteria, maybe you can get down to those one or two different compounds that nobody had really looked at before and see if they also have antibiotic activity, and then see if they're maybe hitting the same pathways that other drugs are hitting. And oftentimes they will, and then sometimes they don't, and there really is new activity.
So is there anything else in your reporting that you stumbled across or dug up that you didn't make it into the story but you think is really fascinating, that you wanted to talk about but couldn't?
Yeah, absolutely. I maybe use like a throwaway sentence for this in the story, but robotics is actually a really big deal right now, especially when it comes to synthetic biology and even just like genomics, library preparation, DNA extraction. For the longest time, like it took a physical person with a pipette to go in and move different chemicals around, grind stuff up, go through the process of getting to an outcome in the lab. And this is like a really repeatable process, but it's slow, right? Especially when you're asking a person to keep straight a 96 well plate of samples and pipette a little bit of, liquid into each and every one of those samples in the right order, in the right time frame, and then hope it's done right so that you reach the desired outcome. But now we have liquid handling robots that can do all that for all intents and purposes instantly, very quickly, can do all of this stuff in a repeatable way, in a way that is going to work. And that has rapidly accelerated just the amount of testing that can be done when it comes to screening a compound for effectiveness against like a bunch of different targets. Or maybe like you've modified a compound in 100 different ways and you want to test all of those modifications against a bacteria to see which one does best. All of that is possible to do in a really time-efficient manner now because of robotics.
So I see these systems when I go to a lot of different scientific conferences. How widespread are they in the lab, like out into academic labs? Like I assume Big Pharma has fleets of these things, but for the more academic researchers, do they have the robots yet?
They're expensive. I have definitely seen folks who are like starting brand new labs and they get their startup package in academia, decide like, all right, you know what, let's spend money on these robots. Certainly more established labs like the bioprospecting experts, the synthetic biology experts that have been doing this for a while have gotten the grant funding to invest in these machines. But also a lot of times robots like this are shared resources, right? So one chemistry department in middle of nowhere, Tennessee, just to say something, that might not have the resources of a really high-end R1 institution that's pumping out major research all the time. One robot can be shared by a dozen different labs that might all need to use it for one purpose or another. So I think they're available enough to be really useful for a lot of people, even if it's not something that's necessarily sitting on the desktop of every biochemistry wet lab, right, around the world.
And should we think about the robots, as doing the pipetting for you? Or, I know that they have systems that can do all sorts of complicated manipulations, but are they actually being used for, the medium volume preparations and some other things like that? Or is it pretty much the pipette robot that's winning here?
There are definitely a bunch of other different kinds of robots that are doing some really interesting stuff. I think that the pipetting robot, for the purposes of discovering compounds that a bacteria are producing or synthetic biology, is definitely like the big ticket item that's helping a lot. Mm-hmm. But anything that gives a person more time to do the work that only a person can do is accelerating the rate of discovery for all of these things. Another point that didn't quite make it into the story in a way that represents just how important it is a lot of these language learning AI models are really great for background research and literature review. A graduate student can use these tools to screen thousands of different academic articles in a really short time to maybe get at, well, we have a compound that's really interesting, but we don't know what its function is. Can I use AI to figure out if somebody in the world has published on something similar that might help us get a better idea of what's going on? Because that is something that is very time consuming for a person to like read through all of these papers and see if it's got exactly the kind of thing you need. And you can ask an AI and it'll spit back, maybe a dozen papers or so that could be in the right area that you want. And then you can go in, find something and work from there without having to like dig through all of this stuff and hope you find something quickly.
It's like the new version of the old graduate school chestnut of 2 to 3 years of hard work in the lab can save you 10, 15 minutes in the library.
Yeah.
And this is like the next version of that where very skilled AI use can get you there even faster, I guess.
And of course, AI for coding too is just like absolutely taking off and making data analysis go much, much faster now. Folks still have to have the skills to verify that like it's doing the right thing. You got to have a basic statistics knowledge. You got to have a basic coding knowledge. But all of that is just making it so much easier to get to a conclusion, like the really important nugget of information you want to find much more quickly.
So one of the larger scale questions in your article is, are we in a new golden age for antibiotic discovery? And my question for you is, how do you know if you're in a golden age in your own subfield? How can you tell? It's easy to look back and say that was the golden age.
Yeah, I think it comes down to a consensus, right? And I don't end the article definitively by saying, yes, we're in a new golden age. I've got a couple people who think we're at the beginning of a new golden age. And then I've also got folks who are saying we can't call it a golden age until we get these things into the clinic, right? Because discovering a compound does not mean that the compound actually turns into a real and useful antibiotic. So maybe it is 10 years from now when some of these compounds make their way into the clinic where we can really think about a new golden age starting. One thing I'll say is I had some sources give really great analogies that set the whole thing up. Jerry Wright, who's faculty member at McMaster University, who actually discovered this lasopeptide antibiotic variocidin and said that we're maybe at the start of a golden age in antibiotic discovery. He says that like finding all of this stuff is a lot like the gold mining operations that take place in his native Ontario. Like we're mining the genomes of microbes to find these new compounds. Honestly, all that comes together as a really cool angle to like dive into the story with. And we've even got some art of like old school like gold miners, but they're looking through fungi. Thank you, Maddie and the C&EN art team for that great illustration. Yeah, so a coincidence and a good analogy can be the foundations of a feature story. And I had no idea this was going to be the cover when I wrote it. Like, I actually found that out, like pretty late into the process of me writing it. And it was like, oh, crap, I better go and do some more reporting. Because it was going to be just a pretty small story. But yeah, here we are now. Super proud. It's my first cover story.
Congratulations.
Hoping to have more soon.
Max, thanks for diving deep on this with us.
Yeah, thank you, Craig. It's been my pleasure.
You can find me on social media as @CraigOfWaffles. Max, how can listeners get in touch with you?
Yeah, the best way to contact me is by email. That would be m_barnhart1@acs.org, or go to cen.acs.org, our website. Look up my name and my contact information is there.
So you can find Max Barnhart's cover story about mining for microbials on C&EN's website or in the June 23rd print issue of C&EN. We've put a link in the show notes along with the episode credits. We'd love to know what you think of C&EN Uncovered. You can share your feedback with us by emailing cenfeedback@acs.org. This has been C&EN Uncovered, a series from C&EN Stereochemistry. Stereochemistry is the official podcast of Chemical and Engineering News. Chemical and Engineering News is an independent news outlet published by the American Chemical Society. Thanks for listening.